Every minute, 4 babies are stillborn globally. This is about 5,000 babies stillborn every day. In the United States alone, this tragic loss of life affects 10,000 families every year. This tragedy might even be triggering for some of us in the audience. Stillbirth is not only personal, but a public health crisis requiring immediate action through multidisciplinary research. My name is Tsegaselassie Workalemahu. I am an assistant professor of obstetrics and gynecology here at the University of Utah School of Medicine. It's a great honor for me to speak to you today about expanding novel genetic research to address stillbirth, a silent epidemic. First, I would love to tell you about my atypical but fascinating career journey that brought me to a wonderful place like Utah to be a maternal fetal medicine researcher. My journey began with a love for mathematics. Unlike many in medicine, my first degree was in math from a liberal arts college in Pennsylvania. Many thanks to my late dad and my living mom who taught me critical thinking skills, but also ability to link abstract things like math to solving practical problems or helping others. I've always been profoundly fascinated by mathematical modeling to solve real-world problems, so much so that I went on to pursue a master's in applied math at Georgia State University. There I met my mentor, Dr. Marina, who introduced me to a growing field known as bioinformatics, which intersects biology, statistics, and computation. This field really fascinated me, and it instigated my interest in statistics in public health. After my graduation, I was employed at Harvard School of Public Health, where I was tasked to support large public health databases through bioinformatics and statistics. So quite fitting to what I wanted. At Harvard, I met renowned public health experts, Dr. Michelle and Dr. Daniel, who kindly mentored me and inspired me to pursue an academic career and introduced me to numerous individuals working on genetics of complex diseases in populations. So at this juncture, I had a rewarding opportunity to contribute today and work with a lot of people around the world to contribute to our understanding today about why we are different from one another in terms of our height and weight and what determines the biology of these phenotypes. While these were pioneering, helping us understand complex issues like obesity in adults, to me it wasn't impactful enough. To me it made sense to understand the early development that to understand how diseases manifest in adults. In other words, imagine if you had the opportunity to intervene early in development and optimize the health of the future populations. These are complex but completely transformative research questions that intrigued me, but they require specific training in study design and maternal child health epidemiology. So I needed further training, and I went on to pursue a PhD, and I obtained my PhD in epidemiology from University of Washington, and a postdoctoral training from the NIH Child Health Institute. Many thanks to my mentors at the time, Dr. Gale and Dr. Fasil. I was inspired by this idea that pregnancy can be seen as a window of future health. This very idea that by preventing pregnancy complications, we can improve the health of the mother and the offspring throughout their life course, hence the future population. That's why I dedicated my career to maternal fetal medicine, and I embarked on an exciting journey to the University of Utah to conduct research on pregnancy complications like stillbirth. What is stillbirth? In the US, we define stillbirth as pregnancy loss after 20 weeks gestation. It's a silent epidemic that occurs far too often. What's interesting is that as we get more and more advanced with technology, we're finding genetic abnormalities. But it's disheartening that at least a quarter of them remain unexplained, meaning that families are sent home without knowing why stillbirth occurred. As you can imagine, this has led to a lot of frustration for families looking for answers. Even with the overwhelming grief and impact stillbirth has on a family, it's largely understudied. It turns out that the placenta holds lots of clues. The placenta is this fascinating organ that has a very short lifespan, whose sole purpose is to support the development of the fetus and gets thrown away after birth, seems to be important. You might be surprised to know that the placenta is mostly genetically distinct from the mom. Since we know both the mom and the dad contribute to the genetics of the fetus, the inherited risk factors are important, but they weren't fully investigated. The good news is that here in Utah, we have a unique resource of family structured data. One of the attractions about Utah for genetic epidemiologists like me, we wanted to investigate family relationships, both maternal and paternal side, and ask, is stillbirth familial, or does it aggregate in families? And what we found using this data is that stillbirth runs in families. There is actually inherited risk coming from the dads, indicating the importance of including both mom and dad in stillbirth research. In fact, there was a missed opportunity when only looking at maternal risk factors when we know there is genetic risk passed down from the dad as well. We were the first to leverage a rich data source like this to understand the inheritance pattern of stillbirth. This is important because it provides the justification to investigate inherited genes that may be causing unexplained stillbirth. And of course, you might be asking, what have we done to investigate those genes? So to address this, we sequenced the mom, the dad, the placenta from the stillbirth, and the live births, and compare the genomes across and within to identify genetic abnormalities. And using only few sets of families like this, we were able to detect genetic abnormalities in unexplained stillbirth. However, we need more families to participate in our genetic research to identify causal genes that will be impactful for clinical guidance. Why is this important? Well, there are many families who are left to wonder what happened, and knowing the genetic causes will help with closure and bereavement for these grieving families. And with that information, we can not only optimize the health of the babies born too premature with neonatal conditions, like you heard from my colleague before, we can also inform guidelines for developing therapeutic options for human survival and prevention of stillbirth. Certainly, this is a task that requires all hands on the deck. We are a multidisciplinary team that have joined hands to address stillbirth from a multidisciplinary angle. And your joining hands and partnering with us, either through research participation or philanthropy, will bridge the gap between the silent grief of countless families and the hope offered through advanced genetic research. Together, we can turn the tide on stillbirth, offering not just answers, but a legacy of life-saving knowledge and healing. And I would like to thank my team members who've partnered with me in expanding novel genetic research to stillbirth. And I would like to thank my family and the mentors I mentioned throughout my slide, who I call life coaches, and my funding organization, and especially families who are participating in our stillbirth genetic research. And finally, I'm providing here a link to, for those of you, or for those you know, might be grieving, and these are some of the resources. Thank you very much.